Sequence analysis could not identify a TF2B homologue in polymerase I transcription. However, structural analysis revealed TAF1B, a subunit of the SL1 complex, shared many domains with TF2B. TF2B contains a zinc ribbon domain, which facilitates polymerase II recruitment. A similar domain in TAF1B suggested it may also be involved in the recruitment step. Cyclin-like folds in TF2B interact directly with DNA. Equivalent structures in TAF1B implied a role in binding the polymerase to the DNA strand. Disparities between the proteins were also observed. Highly conserved domains in TF2B's connecting region, known as the B linker and B reader, were absent in TAF1B. Although TAF1B's connecting region is active in transcription, these domains were not observed. The Tata box binding protein is not used in polymerase I promoter binding. Instead, TAFs, such as TAF1B, fill this role. Although TAFs are present in the polymerase II assembly, they are associated with the transcription factor TF2D and not TF2B.
Despite similarities, a similar function could not be deduced based on structure. Mutagenesis experiments were devised to determine the role of TAF1B.
We thought if it's a zinc coordinating amino acids, then if we mutate those, we thought we might mutate a zinc finger and therefore not have DNA binding. But it wasn't a zinc finger, it turned out to be a slightly different variant that also is involved in coordinating zinc ions. But the mutations gave a very dramatic phenotype, and yet we couldn't pin it down to any of the obvious defects in assembly of the complex, recruitment of the polymerase, DNA binding. They were all fine. And it turned out that actually there was a defect after all these events happened. So it binds DNA fine as a properly assembled complex. It recruits the polymerase, but then there is a problem.
It was found that TAF1B was not essential for polymerase I recruitment. It appeared that its main function may occur post-transcriptionally. This is similar to polymerase III transcription factor, BRF1, a TF2B homologue.